Hello friends, in this video we are going to discuss about ribosomes, cell organelles in detail. First of all, its definition. Ribosomes are one of the most important cell organelles that are found inside the living cells that produce proteins from amino acids during a process called translation. Translation is also known as protein synthesis. It is described as cells' protein factories. These are complex molecular machines. Ribosomes are specialized cell organelles that are found in prokaryotic and eukaryotic cells. These are required by every living cells. Now the history of ribosome. Ribosomes were first observed by Robinson and Brown in the plant cells in 1953. It was first observed by Palade in animal cells in 1955. Occurrence. These are ubiquitous in all cells. These are found in prokaryotic as well as eukaryotic cells except in mammalian RBCs and mature sperms. In prokaryotes, it is present freely in cytoplasm. In eukaryotes, it is either present freely or are embedded on outer surface of rough endoplasmic reticulum or nuclear envelope. Matrix of mitochondria and stroma of plastids also contain ribosomes. It is present in large numbers in cells actively synthesizing proteins and present few in starving or less active cells. Now the structure. These are mostly oblate or spheroid in shape. In prokaryotes, its diameter ranges between 17 to 21 nanometer and length ranges between 20 to 29 nanometer. In eukaryotes, diameter ranges between 20 to 24 nanometer and length ranges between 30 to 34 nanometer. Functional ribosomes consist of two subunits, large subunit in short L and small subunit in short S. It is also referred to as ribonucleoprotein because it is complex of RNA and protein. It also contains metallic ions like Mg2+ and some amount of lipids. Amino acids are added at bigger subunits and messenger RNA binds and is decoded at smaller subunits. Now let us see different sites of larger subunit. First is P site or peptidal site. It is meant for attachment of the peptide chain carrying transfer RNA. So transfer RNA which carries peptide chain attaches to P site. Then comes the A site or amino acid site. It is meant for attachment site of charge transfer RNA. Charge transfer RNA which carries activated amino acid attaches to A site. Then comes the E site. It is meant for attachment site of free transfer RNA after it has transferred the peptide chain. So transfer RNA after it has transferred the peptide chain is free, which then attaches to the E site. Now two subunits exist separated from each other in the cytoplasm and combine only during protein synthesis. This is the larger subunit and this is the smaller subunit of the ribosome. These are the E, P and A sites of larger subunit. This is messenger RNA which is decoded or translated into protein. Now let us see types of ribosomes. There are two types of ribosomes, 70S and 80S. 70S ribosomes are found in prokaryotic cell. These are also found in mitochondrial matrix and stroma of plastids in eukaryotic cells. 80S ribosomes are found in cytoplasm of eukaryotic cells or outer surface of rough endoplasmic reticulum and nuclear envelope. 70S ribosomes are smaller in size. 80S ribosomes are comparatively larger in size. Molecular weight of 70S ribosome is 2.7 into 10 power 6 Daltons. Molecular weight of 80S ribosome is 4.5 into 10 power 6 Dalton. In 70S ribosome, larger subunit is 50S and smaller subunit is 30S. In 80S ribosomes, larger subunit is 60S and smaller subunit is 40S. In 70S ribosome, Mg2+ helps in subunits to unite. In 80S ribosomes also, Mg2+ helps in subunits to unite. In 70S subunit, three types of RNA are 23S ribosomal RNA, 16S ribosomal RNA and 5S ribosomal RNA. In 80S ribosomes, 4 types of RNA are present: 28S ribosomal RNA, 18S ribosomal RNA, 5.8S ribosomal RNA and 5S ribosomal RNA. In 70S ribosomes, 23S ribosomal RNA and 5S ribosomal RNA are present in 50S subunit and 16S ribosomal RNA are present in 30S subunit. In 80S ribosomes, 28S ribosomal RNA, 18S ribosomal RNA and 5.8S ribosomal RNA are present in 60S subunit and 18S ribosomal RNA is present in 40S subunit. In 70S ribosomes, 30S subunit has approximately 21 proteins and 50S subunit has approximately 34 proteins. In 80S ribosomes, 40S subunit has approximately 30 proteins and 60S subunit has approximately 50 proteins. In 70S ribosomes, 65% RNA and 35% proteins are present. In 80S ribosomes, 45% RNA and 55% proteins are present. So we are talking about 70S and 80S. What this S stands for? S is Swedberg unit. It denotes sedimentation coefficient. It is a measure of how quickly a particle can sediment in an ultracentrifuge. Greater S value means larger and denser the particle. Lesser S value means smaller and lighter particle. Now the important characteristic features of ribosomes. These are smallest submicroscopic ribonucleoprotein particles, also known as pallid particles or cell engines. These are membraneless structure. These are primarily involved in protein synthesis, hence known as protein factories. Eukaryotic cells have higher number of ribosomes compared to prokaryotic cells. Ribosomes can exist alone or in groups known as polyribosomes or polysomes. In polyribosomes or polysomes, many ribosomes are linked by a long string of messenger RNA. Now the functions of ribosomes. These are responsible for producing biological proteins. Primary function of ribosomes is the formation of proteins from amino acids known as translation. During translation, hereditary information from messenger RNA is translated into proteins by ribosomes. Proteins that are found in cytoplasm is synthesized by free ribosomes and are used within the cytoplasm. The bound ribosomes proteins are transported outside the cell. Peptidal transfers and peptidal hydrolysis are two critically important biological processes in which ribosomes act as catalyst. So this is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.